I'm Ryan Continente, and I'll be presenting Forensic Analysis of Counterfeit Psychotropic Drugs by the Guardian et al. 2024. Let's start. The agenda that I'll be presenting is divided into five parts. First, the introduction, material and methods, results and interpretation, discussion, and conclusion. The counterfeiting of pharmaceutical products has been an increasing problem for the last decades worldwide. As counterfeits come in many forms, also the definition of counterfeit medicinal product differs in various countries and sectors. What is a falsified medical products? According to WHO definition, it corresponds to medical products that deliberately, fraudulently misrepresent their identity, composition, or source. Criminals take advantage of the population needs, for instance, in terms of vaccines, COVID-19 treatments, and the most targeted by the counterfeiters are the central nervous system pharmaceuticals like psychotropic drugs. As per the researchers, none has been dealing with the counterfeiting of psychotropic drugs using a combination of several tools, both classical and non-conventional ones. In this study, another strategy will be presented based on high-performance liquid chromatography, 3D microscopy, near-infrared spectroscopy, imaging tools, gas chromatography with mass spectrometry, nuclear magnetic resonance, and X-ray fluorescence spectroscopy. Finally, the forensic analysis of the counterfeits will provide indications on possible links between the cases, which might be very useful for law enforcement and prevention purposes within the companies. The packaging in which the counterfeits were seized is out of scope of this study since most counterfeits were encountered in loose plastic bags. Three batches of the genuine medicine named G1, G2, and G3 were chosen as references. For each batch, 1 to 10 tablets have been analyzed with a particular analytical method. Then seizures of counterfeits of the psychotropic drug named C1 to C10 have been studied in this article. These cases had all been confirmed as counterfeits by the time of this research, and the tablets were remeasured for the purpose of this study. Depending on the chosen analytical technique, 1, 10, or all the tablets have been analyzed. Non-destructive methods have first been used for all the tablets, and after controlling the sampling, was homogeneous. Only 1 to 10 tablets have been analyzed depending on the instrumentation. The counterfeited tablets were first compared with the genuine references using the Videometer Lab 4 spectral imaging system from Videometer to study the surface color, texture, shape, and indirectly have an indication whether the chemical composition was similar to the genuine tablets. Also, this technique was chosen since all tablets can be analyzed in a non-destructive way and is very fast. All the received counterfeit tablets and 10 reference tablets per batch have been analyzed, and a mean spectrum was generated after integration of the surface of the whole image and a mean standard deviation applied. The visible spectra were overlaid to compare the counterfeits with the references. As for the 3D microscope, the depth of the embossing on the counterfeited tablets and the genuine tablets was compared, and the choice for this method was driven by the fact that it is non-destructive, fast, and can give a first information whether the tablets are counterfeited or not. For near-infrared spectroscopy, potential differences in the chemical composition and some physical characteristics of the tablets were then evaluated by NIR spectroscopy in a non-destructive way. 10 tablets per counterfeit seizure and 10 tablets per genuine batch have been analyzed with this method. The spectra were pre-treated using a standard normal variate, and a PCA was applied on the spectra as an exploratory tool so that the differences and similarities between the spectra were enhanced, allowing the detection of underlying clusters and providing some information about the homogeneity of the samples. To confirm the results obtained by NIR imaging was then tested. NIR imaging systems enable the acquisition of NIR spectra for compound characterization on a large area of the samples for their spatial localization. This technology has had several pharmaceutical applications, such as the detection of counterfeits. Then it was followed out by X-ray fluorescence, where it identifies potential differences in the elemental composition of the counterfeit tablets in comparison with the reference tablets. The whole range of elements from sodium to uranium was chosen. One tablet per counterfeit seizure and per reference batch has been measured on both sides, and for each side of the tablet, three measurements at three different places were performed. The HPLC methods in the sample preparation described in the study were previously developed for routine analysis of the genuine medicine. Since the technique is destructive and according to non-destructive methods, the sampling within the seizures was homogeneous. Only three counterfeits per seizure have been analyzed. Since only a few tablets were available per seizure, the analytical method had to be adapted accordingly and the matrix effect evaluated. Differences in the excipient's profile of the counterfeits in comparison with the one of the genuine tablets have been investigated by GCMS. In order to confirm the results obtained with previous non-destructive methods, several tests were performed to evaluate the resolution and reproducibility of the chromatographic system. They also included preparation without derivatization and preparation with derivatization. NMR is a technology based on the magnetic properties of certain atom nuclei, such as hydrogen, therefore called 1H NMR, which is the method proposed. The chemical shifts were reported in parts per million, and for all the spectra, the reference position was adjusted to the residual proton signal of the DMSO solvent, and a data processing consisting in a baseline correction was then applied. Results and interpretation, authentication and determination of the chemical composition. Results of the spectral imaging analysis of the counterfeit seizures was compared to the references. One mean spectrum was computed for one seizure of suspected counterfeits, that is in green, and the references in red. A PCA was then applied on the reference tablets and on the counterfeits, revealing some differences between artificial colored images of the counterfeits and the references. To investigate the physical aspect of the tablets, the depth of the embossing was measured for each tablet along the perforation of the brand name. The same starting and ending points have been chosen for all the tablets. The whole formulation of the tablets and indirectly their physical characteristics have then been studied by NIR spectroscopy. The samples were overlaid in a PCA computed on the reference and counterfeit spectra. Slight differences can be observed between the spectra of counterfeits and genuine products at around 5,100 reciprocal centimeters. This corresponds to potential differences in the OH bands, which might suggest a different sugar composition between the counterfeits and the references. It can be moreover be deduced from the spectra and the PCA that the counterfeit tablets are quite homogeneous within the seizures. Therefore, based on the physical aspect of the tablets, the counterfeit tablets can be differentiated from the genuine ones along with the spectral imaging. NIR spectra of the suspected counterfeits in blue and of the references red after SNV or standard normal variate pretreatment. PCA obtained on the reference and counterfeit NIR spectra was pretreated with SNV. The loadings show that the distinction is mostly made around 5,100 reciprocal centimeters. For a clearer observation of the differences between the references and the counterfeits were all represented by the same color. Slight differences had been observed between the NIR spectra of counterfeits and the references. The samples were therefore also measured by NIR imaging in order to add dimension to the analysis. Near-infrared live images first show differences in the glowing, the granulation, and the distribution of compounds. The similarities between the references and the counterfeits could also be detected on NIR images, for instance, at 1930 nanometers on images treated by normalization, and additionally at 1515 nanometers and 1700 nanometers on images pre-treated by normalization and first derivative. The similarities between the references and the counterfeits could also be detected on near images, for instance at 1930 nanometers on images treated by normalization, and additionally at 1550 nanometers and 1700 nanometers on images pre-treated by normalization and first derivative. These differences can mostly be attributed to differences at the OH bands, as previously detected by near spectroscopy, and also at active ingredient bands. An elemental analysis was performed with X-ray fluorescence spectroscopy on the reference and counterfeit tablets in order to detect potential differences in the elemental composition between the counterfeits and the genuine tablets. The test revealed an amount of magnesium at least twice lower in the counterfeits than in the genuine tablets. No other significant difference could be detected either on the spectral overlay of the references and the counterfeits or the mass percentage of other elements. HPLC tests were performed for the analysis of the active pharmaceutical ingredient. The correct active ingredient was identified in all the studied counterfeits, and the same content was detected as the genuine tablets. In addition, three degradation products of the active ingredient were found, such as for the original active ingredient. Therefore, according to this HPLC method, no differences could be detected between the counterfeits and the genuine tablets. No difference could be detected between the counterfeit tablets and the references during the test performed without derivatization. However, the measurements after derivatization revealed a different proportion of sugar at retention time between 12.4 and 13.5 minutes between the references and the counterfeits, as well as a different proportion of palmitic acid and stearic acid. Example of counterfeit C1 is presented on as the gas chromatogram overlay obtained after derivatization, the reference G1 in blue and the counterfeit C1 in pink. 1H nuclear magnetic resonance analysis has been performed on the counterfeit tablets to confirm the small formulation differences with the references obtained by GCMS. The 1H spectra revealed different proportions in the excipient composition. Indeed, signals of higher intensity can be attributed to palmitic acid and stearic acid, were detected for the counterfeits at approximately 2.26 parts per million, 2.72 parts per million, and 5.4 to 5.5 parts per million, as previously suggested by GCMS. Since these signals were below the limits of quantitation, the different proportions of excipients could not be quantified by QNMR. An example of spectra is displayed on the next slide. An example of spectra is displayed on Zoom 1 proton NMR spectra of two references, below in green and red, and of counterfeits C1 and C2 above in purple and light blue. The counterfeits studied in the article all originate from seizures that were made in the same region and within the same calendar year. Apart from the similarities found with the seizure information, the analytical data generated during the presented study contribute to the detection of links between the seizures. The batch-to-batch variability of the counterfeits is not comparable to the one of a GMP or good manufacturing practices in legal pharmaceutical production environment. Therefore, the composition of the counterfeits might differ between the studied seizures. Despite this fact, some similarities were observed between the physical parameters of the counterfeit tablets. In this article, the analysis of counterfeit tablets presenting a chemical composition close to the genuine tablets has been proposed. The purpose of this study was to present the results of analysis performed with several technologies so that differences between the present counterfeits and genuine products might be highlighted. Non-destructive methods have first been used. The spectral NIR imaging and 3D microscopy help consider physical differences between the genuine tablets and the counterfeits. It enables counterfeited tablets analysis, detailed compositional information, and comparison without sample preparation, with references gathering both chemical and physical dimension. NIR is indeed a good method for the analysis of sugar in tablets, while differences in the proportion of palmitic and stearic were identified by both NMR and GCMS, linking to the different proportions in the magnesium, which was detected by the XRF spectroscopic method. HPLC proves that the analysis of the whole formulation is necessary when dealing with the authentication of medicines. And here are the additional notes that were included in this study. For continuation and conclusion, counterfeit medicines present a danger to the patients given their unknown condition. It might not contain any active ingredient. Some are quite close in composition to the genuine products. It is important to follow a correct analytical strategy so that all counterfeits can be detected. Analysis of counterfeits of psychotropic drugs has been presented using several chromatography, spectroscopy, imaging, and elemental analytical methods. Analysis of the excipient's formulation is necessary, and the analytical data that are generated are of great help for forensic intelligence. The references of the study. Thank you for listening.